Bonjour, everyone. Je m'appelle Kaylee,
Et je m'appelle Selena.
and today we're here to teach you how to use a Foldscope microscope. So a microscope is a powerful tool that's used in science, and it allows you to see small specimens very, very, very up close. So basically, it enlarges an image in real life so that you can notice smaller details. And we are going to be teaching you how to use a special type of microscope today called the Foldscope. So Selena and I and everyone on our team donated a batch of Foldscope microscopes to your school. And we'd like you to take a second to locate your Foldscope, which you'll be sharing with a few people at your table. And so if you'd like to pause this video while you take care of that, feel free to do so. All right, welcome back. Hopefully you each have your one pouch of Foldscope microscope right in front of you or right in front of your table if you are sharing with a couple of your table mates. So at this time, we'd like you to take your pouch, and yours might be a different color than ours, and take the zipper and unzip it. So Selena will show you how to do that. Now remove the Foldscope microscope out of the pouch. Yours should also be blue like this one. And this is the microscope instrument that is going to be used during all your interactive experiment lessons that we'll be doing with you. All right, so just take a second and set your Foldscope aside. And now let's take a look at some of the other things that are found in your Foldscope pouch. So Selena's gonna take a second and remove each thing one by one. So first you pulled out your Foldscope field guide, and this is just an example of a few different specimens that you might see under your microscope. So that's like protozoa, algae, little bacteria, and other microorganisms that are too small to see with the naked eye. Next, she pulled out a kit of smaller different instruments that we use for different specimens, but we'll talk about that on later lessons. That doesn't concern us for today. This is very important. This is the light. So every microscope needs a light because it illuminates the specimen and allows you to see different angles and different colors, shades, and shapes of your specimen better. So this is the light, and it's very important. These are your slides. So the one that Selena's showing in front is the pre-prepared slide, and that is called the fern rhizome, and that's a special type of plant. But behind this slide are two other slides that are completely blank and completely clean and sterilized, and these are the ones that we'll be using for our future experiments. These are clear slides that you'll use on future experiments, but again, not for today. This is an extra shiny slide that Foldscope wanted to include. You can take this home if you'd like, but it's basically just a small gift inside the Foldscope package. These are called forceps or tweezers. Selena will show you how they're used. Basically, you just pinch them and they can help you pick up small objects when you're out finding different specimens in the field. They're very helpful because sometimes your fingers are too big to pick up smaller items. This is an Eppendorf tube. Ours is blue, but yours should be clear. And basically you can put different liquids or specimens inside of this tube. And lastly, this is your pipette. Yours might have two pipettes, but most of them should have just one. And these are coupled with the Eppendorf tube, and that's how you transfer liquid from one place to the tube.
Can I show one?
Oh yeah, sure.
Okay, so I'm gonna show you guys real quick how you might do it. I'm not sure if your tube will come with liquid, but when you do use it for experiments, you'll have some. So you wanna squeeze the pipette first like this, then you dip it in here, then you're gonna let go of the pipette and see, now you bring it up and I've collected some in here. And so when you wanna have it come out again, you just squeeze it and then it comes back through here. like that.
So really cool and really simple and really efficient for scientists. All right, so now let's learn how to prepare and use your Foldscope. So for today, we're just going to be using the pre-made slide of the fern rhizome that's in your slide kit. But in the future, we'll actually be preparing our own slides with real life specimens. So take a second to put everything back in your kit because we want to make sure that we don't lose anything because everything is very important for future lessons. So go ahead and do this with me. And you can just leave out the slides, the light, and your Foldscope microscope. And you can put your kit to the side. All right, and now we're gonna just be transferring to a clearer camera for you guys. All right, so here is your Foldscope microscope. As you can see, it's made of a kind of thick, glossy paper. In English, we call this cardstock. So yours should look like mine. It should already be made and assembled. If not, we can send you some assembly instructions in French. First, what you're going to do is you're going to take the slide kit in plastic and you're going to open it up like this. And you're going to remove the slide that is called fern rhizome. And you can see there's a small purple dot in the center. Now what you're gonna do is you're gonna take your Foldscope microscope and you're gonna take this yellow flap and open it up. It should open up pretty easily because it's just a magnet. Since your microscope is brand new, it's gonna have a white paper with red tabs on the side. Just pull the red tab out and it should come out just like a slide. And it should look empty just like this. It's very simple. All right, so now you're going to see how you insert a slide into your Foldscope. So very important, you're gonna take the slide right here and you're going to insert it into the Foldscope face down. So the writing should be down. It should not be facing up. It should be facing completely down. You're going to, this side is better. You're going to insert the slide from the small pouch. And if you can see, let's see if Selena can position the camera. Yeah, you can kind of see that there is an arrow right here. And this arrow indicates that you're going to slide the slide right through here. So make sure that the slide goes directly through the arrows. Just like that. So now you can see my slide is coming through that arrow right here. And we're going to do the same thing through the other side. So there's an arrow right here. Yeah, you can see it very clearly on our end. And you're just going to take your slide and push it right through there. So now you guys have inserted your very first slide into your Foldscope. And you're going to want to make sure that your specimen, in this case, the purple circle, is aligned directly with the lens on the Foldscope. So as you can see, the two black circles are aligned together. Now what you're going to do once the two black circles are aligned is you're going to close your Foldscope. It should close with a click, just like a magnet. And you're going to take your light. Like we said earlier, the light is very important because it illuminates your specimen and it allows you to see it better. Your light is going to have a small plastic tab sticking out and you're just going to want to pull it. Since mine isn't brand new, it doesn't have it, but just pull your plastic tab and it's going to activate your battery. Once you have pulled the plastic tab, turn on your light and you can tell it's on just like this. This part is going to be illuminated. Once you have turned on your slide, attach it onto this end, just like a magnet. You see now that I'm not touching it anymore. My hand is over here. So it's been attached via a magnet and it's really cool. All right, now what you're going to do is you're going to flip your Foldscope and face it towards yourself, towards your face. And let me show you a couple features of the Foldscope that are going to come in handy for future experiments. So you should hold a Foldscope like this with your 2 thumbs on each circle point. So thumb here, other thumb here. And with these thumbs, you're going to shift the Foldscope left to right. And this is going to help you find and locate your specimen in case your slide has moved around. You can also shift it not only left to right, but up and down, up and down, right, left, up, down. So quite simple. So now that you've learned how to move your specimen, you're also going to want to learn how to focus your specimen. So oftentimes in a microscope, the first image that you see is not going to be crystal clear. In fact, it might be a little bit fuzzy or a little bit blurry and hard to see. So in that case, you're going to want to focus your specimen, and you're going to do that with this tab right here. It's a little bit lighter shade of blue, and it has these ruler markings on it. So each microscope is different, but you're going to want to focus it by moving this left to right. And you'll be able to see once you look inside of the lens, which way focuses it better or not. So I'm going to take a second to focus my microscope, and I invite you to do the same by moving, once again, this white tab left to right. And once you've done that, I'll show you my specimen and we'll see if yours matches mine. So Selena's going to show you how I put mine up to my eye, and I'm trying to find the purple specimen in here. Here it is. I can tell it's really blurry. So now I'm going to take the microscope and I'm going to focus it with that white ruler tab that I showed you at the beginning. For me, I just had to move the white tab a little bit to the right, but not too much. And now I'll show you my point of view, and hopefully yours matches this. There you go. So this is exactly what I'm seeing inside of my microscope right now. It's a lot of small circles that are very nice and colorful, but this is actually a lot of different cells that are in the fern rhizome. So each circle is a cell, and within each circle, there's a smaller purple dot, and that's the nucleus of the cell. So you'll learn about that in a future lesson, but it's basically the brain of the cell, and it's big enough where you can see it with a Foldscope microscope, which is pretty cool. So yours should hopefully match my point of view, which is what you're seeing right now on the video. If not, take another second to just play around with the Foldscope a little bit, maybe move these left to right, up and down, or focus it a little more. You can even open up your Foldscope and make sure that the purple center is aligned right over the lens, just like this. And if not, you can move it a little bit. But other than that, that is a quick lesson on how to use a Foldscope microscope. It's a very powerful instrument and it's very special. So make sure you store your Foldscope very carefully in your pouch and make sure that it's easily accessible for future lessons so that we can learn and use the Foldscope together. So other than that, I want to thank Selena for helping me with this Foldscope tutorial, and I want to thank you guys for listening. And once again, if you have any questions, just let us know. Thank you. Goodbye.